Curiosity. Kill the rat. Curiosity. Kill the rat.
Hello, everyone, and welcome back to another episode of Curiosity Killed the Rat. Today, we are in our second part of our very special Pint of Science special. I am Matt. I am not a scientist, but I love science. I love learning about science. I love all things science. I am also, of course, joined by my lovely co-host, Kate.
Sup, guys? Yeah, I am, I guess, the normal scientist of this show, but today I am not the scientist as the same as last week. I am not the key scientist that we are going to, because we have been lucky enough to be paired with the lovely Pint of Science crew. So if you haven't heard of Pint of Science, it's this awesome festival that normally happens live in person in those first, that first weekend or so of May, where scientists go to pubs and share their research, talk about their stuff. As we are well aware, pubs are not a thing right now. We're in the middle of a bit of a cough on cough crisis pandemic thing.
I mean, if you're going to cough, make sure to, cover your mouth at least. So wash your hands afterwards.
Coughing into my elbow. I know you guys can't see the Zoom call that Matt, Luke, and I are in, but I'm coughing into my elbow. Pint of Science. Yeah, so we are lucky enough that Pint of Science has gone online this year, and we are lucky enough to be joined by our second of three guests today, who I will introduce in a quick second. But I just want to quickly say, if you want to find more of the Pint of Science stuff, a lot of the stuff's been put online this year already. You can follow the hashtag PintAU online on Twitter, for example, or you can follow at Pint of Science AU or whatever country you're from. If you happen to not be in Australia, just, Google your local Pint of Science, see what they've chucked online this year. But yeah, like I said, I'm Kate. I'm an addiction neuroscientist by origin, but luckily enough, we have a guest here today. It's super exciting. We have Luke Whiley, who is a researcher at Murdoch University. Luke, how are you? So excited to have you here today.
Yeah, good morning, Chris. Great to be here. It's great to very much for having me on, and looking forward to chatting about the science over the next hour or so.
Yeah, no, super keen for this episode. So as I understand it, I mean, you've already been part of one of the Pint of Science activities online this year, and if people want to hear more about your stuff, they can just chuck in brain health in our modern world, chuck that into YouTube, and you'll find the panel discussion. But super exciting for you to rock up and chat to us today. What are you going to be talking to us about today? What is your area in the great big world of science?
Yeah, sure. So I'm based, I say, at Murdoch, and we're a part of a new centre which was set up last year, called the Australian National Phenome Centre. And here we perform a type of technology called phenotyping, where we actually try to measure all the chemicals that may be in someone's blood or urine, and what's kind of going on in their metabolism at any one point in time. And as part of that, we can do some pretty cool stuff where we try and track these patterns and how they change, and just try and find out what causes disease and what causes people to age healthily or develop dementia as they get older.
So Matt, I'm going to turn to you right now, because I understood a lot of those words, but I know you come in here, same as a lot of our listeners, you come into this discussion with less of a science education. Do you know what a phenome is, as opposed to a genome, as opposed to what questions do you have with that in particular, introductory statement that Luke gave
I don't
us?
know. The only real gnomes I'm familiar with are the ones that sit outside of gardens, and even then, I'm not quite sure what their purpose is. I think it's to scare off birds or something, but I don't know. What I understood from that, I understand that you can, check blood for things and be able to find things wrong with that. I know what the genome is. I think I understand that it's the way that we have something to do with how we've sequenced our DNA and being able to find stuff within the DNA. But I know bugger else more than that. As I've said in the past, biology is definitely my weakest point when it comes to science. So yeah, no, I don't fully know.
So, Luke, do you want to break it down for the lay people among our audience? First of all, at the most basic sort of level, what does phenotyping mean? What is pheno versus genome versus what are these know-me words? Very very basic question, but just give us the elevator pitch.
Yeah, sure. So we inherit our DNA, and we can't do anything about that. We inherit that from our parents, and that is our genetic code. And every person has a very unique genetic code inside our DNA. And the study of that for an individual is called our genome, or our genomics. And with people studying what each bit of that code actually means in real terms. So we might inherit it, but whether it changes the color of our eyes or our height, it's all wrapped in sort of that code, and it's in untangling that. And that is the study of genetics and genomics. And then the next step on is, just because it's written in the code doesn't necessarily mean it's been activated. So when we're reading that code, we have and it's actually transcribed. So then we have something called transcriptomics, and the amount of reading that happens of that code and actually what's done with that code changes from person to person as well, and changes on their environment. So if we're under stress, perhaps we might read a bit more of that code and then express something from that code. So the study of that section of our ome, if we like, is our transcriptome. And then downstream of that, how much of those is actually turned into something like useful for a biological process. And those bits of code are turned into proteins and enzymes and other biological aspects that actually do a job inside the body. And just because you've got the code for that doesn't necessarily mean you'll produce that specific protein to do a job. Okay? So that's what we would call the proteome. And then right at the bottom of that is, if we were to say eat something or take a drug or be exposed to something in our environment, we sort of are always being exposed to chemicals, whether it be sugars or fats or lipids, and they get processed by the body and is part of what's called our metabolism. So we study all of those chemicals, and then we call that the metabolome. Okay, so these omic sciences are very ring-fenced, but together the output of that makes you who you are. So you might have the genetic code, but you also have the environmental influences. You have your diet influences, whether you smoke, whether you drink, whether you do exercise, and all of that combined produces who you are and what you do, and it may affect your personality, it may affect your appearance, but that output is a person's phenotype, whether they are living healthily, whether they have a disease. And then that combination of things together, we can start to spot patterns in it. So maybe one group of people develop a specific disease, but another group of people don't develop that disease. And It's that study of trying to find out why people develop disease. Is it their genes? Mm-hmm. Is it what they're doing in their lives? Or is it actually a sort of a combination of the two that does that?
Nature versus nurture, but in the most scientific sense.
Exactly that, yeah.
So the science of the science of the gnomes and the ohms is the science of just trying to figure out people and person cells and everything that makes a person, from what they were born with to what influences them later in life.
Exactly, yeah.
Right.
Yeah.
Okay.
And it's these little influences that change over time that are likely to contribute to disease. It may not just be one event that happens, but maybe it's 1,000 events happening that just happen to trigger someone's genetic code, and those little changes over time eventually build up and then may result in someone developing a specific disease.
Right.
So my question is, that's a lot of different factors, right, that I'm hearing could possibly influence someone's, phenotype, right? You've got your genes, you've got your environment, but even, you break down your environment, you've got your, physical environment, you've got your mental, emotional environment, you've got all sorts of factors in play. How the fuck does one go about, measuring all of these different factors, controlling for all of these different factors, and then somehow reaching a conclusion about which factors are most important in developing some sort of illness? Like from what I understand, you focus mostly on neurodegenerative disorders such as different dementias like Alzheimer's, for example. But just very generally speaking, how does one design an experiment that controls for every single different life situation that an individual can come across in their life, right? Everyone's lives and their stories are different when you break down person by person. How does one build a study around
How do
that?
you narrow down the variables?
Yeah.
It's a good question. And,
it's a big question, I know, right?
The answer is with a lot of difficulty.
Yeah.
And we're still learning the best ways to do that. One of the ways we can do it is by sheer numbers of people.
Mm-hmm.
If we can get thousands and thousands of data sets, we can actually then start to find really subtle changes that if you only have 10 people, you can't control for the variation between them, because every person is so very different. And there is a certain amount of variation, because like you say, some people may wake up at 5 a.m. and go for a run, but some people may not exercise ever. So how do you control
the
from
difference
that in a
between
small
Matt and I?
Excuse you. Only because it's true.
Cough, cough.
So if we were studying the three of us here, there'd be a lot of variation there from a lot of extremes. But if you turn that into 10,000 people, you can start to sort of subgroup and control for that variation just through sheer weight of data. And then the changes that you do see, you can start to have more confidence that actually it's due to a disease rather than just that person's, the inter-person variation that's naturally happening.
So the bigger your test groups, the better you're able to more easily point to the genetic factor rather than necessarily the independent variables of each person's circumstances.
Exactly. It's almost like you dilute. You have so many people that you dilute that variation out of your analysis.
I potentially have a very controversial question to chuck in here, but at what point do you get concerned that your sample is so big that you're showing patterns that aren't necessarily there? Like that classic, internet comic that every other scientist listening to this has probably seen at one point in their lives where, it shows that, oh, look, green M&Ms are significant because we found them after this many tests. At what point do you decide the balance between this is actually a representative sample or this is the sample of people that's going to show the result that I cough want it to show?
Yeah, we have to be very careful with that. So in the area, we would call models that show that overfitting, in as much as you've looked so many times through the data until you find something. And then you think, I found something. And actually you haven't, because if you look hard enough for something, you can kind of force yourself down that route. So we employ a number of statistical tests that control for that. And has been a lot of work done historically in this to try and avoid that scenario where you stumble across things by chance. If you test enough things, eventually one thing will become significant. So we actually can put some tests into our analysis that prevent that from happening.
So this is kind of almost a follow-up / reiteration of the previous, Pint of Science panel show that you did that people, like I said, can look up if they want more detail. But from that, what I gathered is that your research, specifically like dementia, Alzheimer's, and I kind of want to ask a question around that and what from my understanding, you're looking at, those complex relationships between, our genes, but also our environment, like Matt said, nature versus nurture in terms of Alzheimer's. And I'm just wondering, like, how exactly do you measure this? Because obviously if someone's already been, diagnosed with Alzheimer's, you can't really, I mean, maybe can you study them? Are they of any use, or can you be sure that what you're studying is an effect of Alzheimer's versus an effect of maybe the treatment they've already been put on? Like, how do you actually learn these things about these diseases that, there's all these other complicated factors in terms of, these people have been treated by things, or maybe the healthy population that you're looking at never develops Alzheimer's or dementia. How do you work with these samples and turn it into something meaningful, I guess, is my question.
Sure, and it is a real challenge. So if we go back a couple of steps, to sort of explain, I guess, to the listeners, sort of like the bigger challenge here is that when we come to, if we talk specifically about Alzheimer's disease, the biggest risk factor that we know at the moment of what we actually know is age. So the older you are, the more chance you have of developing Alzheimer's disease. And the next thing that we know is that there are specific bits of our genetic code in our genes that people could inherit that could increase the chance. So it's not a genetic disease as such. If you have that gene, doesn't necessarily mean you're going to get Alzheimer's disease. To complicate matters a little bit, there is a form of Alzheimer's disease that is specifically a genetic disease that if you inherit the gene, you will develop the disease. That typically is quite an aggressive form of Alzheimer's disease, and symptoms start to develop when people are, let's say, middle-aged, so in their 50s, early 50s.
I'm going to just interject with a real quick question. Is there a way to test for this? Is there a way that people can find out if they're, if they have this gene for this early onset Alzheimer's? Or do you have to kind of just wait and see till you get it?
No, so early onset Alzheimer's disease is very well established as the cause for that. The gene is known, and people do get tests for it, but it's a very clear family history. So that is will be well known to families who are affected by that.
Yeah. Okay.
And there is and then there's opportunities for people in those families to be tested. And it's up to the individual whether they then want to have testing, because then there are questions raised about that, whether, would you like to know if you have if you've inherited the gene or not? And that's a very
that becomes a whole
decision.
Big ethical debate, right? You could make a whole Matrix style movie on whether, or Gattaca's probably a better scenario
because that's all gene based.
Yeah, do you need to know? Should you know? Do you want to know? I don't know. That's a whole philosophical debate that we probably shouldn't waste a whole episode talking about. But.
So the picture is a bit complicated because there is this subtype of Alzheimer's disease that is genetic. But if we refer to sort of the Alzheimer's disease that is in the press and people are more familiar with, is sort of the biggest risk factor is age. The older you get, the more chance you have of developing it. But there are some genetic links to that. However, what we don't know is why some people with that genetic risk will develop disease and some people won't. So in this situation, if you carry that gene, you'll have a slight increase of chance of developing the disease, but it doesn't necessarily mean you're going to get it.
It's not a certainty. It's more a predisposition.
So there are a lot of people who have this gene in them, but it's never been activated, so they don't actually end up contracting the disease in their lifetime.
Exactly, and it's a very subtle gene because it's not the full gene that you inherit. So if you think of the genetics as a really, really long piece of code, the risk is only one of those, and the code's made up of what we call bases. There's 4 of them, and if you think of the number of combinations you could get with 4 bases, all in different orders, it can be a really complicated piece of code. And some of these mutations are a single letter in this code. So we call it single nucleotide polymorphism, or a SNP, an SNP, SNP. So it's just one little block or one letter in that code.
But that can have a huge effect.
Yeah, it can have a huge outcome.
Yeah.
And SNPs happen all the time. So the code can go wrong, but it's every so often that SNP, that mutation, results in an actual change in the person's phenotype, if we go back to the earlier in the episode. So we know what these SNPs are. There's been analysis done, there's been work done in massive studies across the world, and analysing the chance of these SNPs and how they affect disease. But what we don't know is, just because you have this SNP, why does that mean you're more likely to develop Alzheimer's disease versus someone who doesn't have that particular SNP?
So can people who don't have these SNPs still develop Alzheimer's?
Yes. So
well, that's just
So just because you don't have the SNP doesn't mean you're going to develop it. But there are also protective genes.
That was going to be my next question. Like, what can people do? Like, what protective factors have, maybe you or just, anyone in the world in this area, like, has been uncovered in terms of, like, people listening to our show being like, oh boy, Alzheimer's disease doesn't sound fun, which you're right, it doesn't sound fun. What do we know? What, quote unquote advice, what has the science shown so far in terms of protective factors?
So some people in their genetic code will have some of these mutations that are actually protective, and people who carry these mutations have a lower chance of developing the disease. And again, it's not really understood why that is. And there's a particular gene which codes for a protein, and it's called ApoE, and it stands for apolipoprotein E. So it's a very complicated word, but it's a protein, it's a protein structure, but it's got it's involved with all sorts of metabolites, because there's fats and lipids and proteins all making this big protein. And there's different subtypes of that ApoE. So there's ApoE 2, 3, and 4. People who carry ApoE 2 are less likely to develop the disease, and people who carry ApoE 4 are more likely to develop the disease.
Interesting.
But there's a lot of work going on at the moment as to why that is. So even though we've spotted that pattern, and there's been lots of studies that have shown that, it's actually not really apparent, or the mechanism of that haven't really been understood as, like, why exactly do those carriers have that effect?
And then what about non-genomic factors? So purely environmental factors. Like, we know, obviously, everyone sort of, or, I'm not going to say everyone, but the majority of people kind of have the association of dementia, Alzheimer's, those are quote unquote old age diseases, right? So we know that age is a risk factor. But, like, what other environmental factors play into increasing someone's risk of developing these neurodegenerative disorders?
Yeah, so that, again, there's been quite a lot of work in the area trying to figure this out. And apart from age, there haven't really been any huge, okay, if you do this, you're definitely going to develop Alzheimer's disease. But there are some things that have been spotted and documented. So there's a lot of work currently going on into cardiovascular health. So high blood pressure and obesity, diabetes are now sort of listed as risk factors. But again, just because maybe a person has been diagnosed with diabetes or high blood pressure still doesn't necessarily mean they're going to go on to develop Alzheimer's disease, but perhaps there's a higher chance.
Yeah.
And one of the challenges that we have to kind of sift through is that we have to kind of group people because maybe they are carrying the genes that are high risk, but they're living a very low risk lifestyle. So if you just look at, let's say, activity, then maybe you can't spot the pattern. But then if you break the pattern down into subgroups, so then you start looking at their genetics as well and their lifestyle, then perhaps then you can start to spot real patterns.
I've just got a question regarding because I know now we're able to fully sequence a person's genome, right? I know a lot of places do it to find out ancestry and that kind of thing. Spotting things like the gene that is associated with Alzheimer's or these other SNPs that are protective against Alzheimer's. When you conduct tests into genes, do you need to be specifically looking for them to find them? Or if you were to just do a generic genome test or whatever, is it able to spot it within that full genome? Or do you have to be specifically looking for it to find it in a person's genome?
You can do both. So you can fully sequence, which takes a lot more time and because the code's huge.
Yeah.
Or you can look for specific SNPs and be more targeted. A lot of the work would be looking for specific SNPs because it's cheaper. You get smaller data sets which are easier to handle. Full genome sequencing is a really huge task. Even computation power to sift through that whole genetic code is huge.
Oh, because it's just that massive.
So people tend to target specific SNPs because it's much easier to work with. And then known SNPs are then associated with disease, and then people tend to focus down that path.
Right. Okay.
So I kind of have one, I don't know, final question, loaded question, the question that you're almost certainly sick of hearing as a scientist, because we're all sick of hearing as a scientist. But for our listeners out there, who maybe our younger listener base, Gen Zers that are here for the memes, that are kind of like, you know what, I don't have Alzheimer's. I'm not going to get Alzheimer's for a good long while. To people maybe, who don't directly have Alzheimer's disease or know someone with Alzheimer's disease, or why is this method of researching stuff, why is this important?
Yeah, sure. No, it's a good question. So the challenge with Alzheimer's disease and to an extent sort of diseases that affect the brain is the brain's a very hard tissue to access and to treat. There's its location's very difficult, and obviously it's a very, very important organ in our body. So, and in addition to that, there's difficulties to get drugs into the brain because we have something called a blood-brain barrier, which is the interface between our brain and our blood. So it's actually very, very challenging to get drugs into the brain. So it's a very challenging disease to study for treatment purposes. Okay, so there's a lot of really great work going on in that area, but it's and there are advances being made, and there is hope, but it's very, very challenging. Now, the other thing that's a massive challenge in Alzheimer's disease is actually how do we diagnose it? So at the moment, diagnosis happens when people start to experience challenges and almost self-raise them. And either their family members may raise changes in behavior or an individual may notice changes in their behavior and memory. And then they'll go and see a doctor and a clinician and they'll get an official diagnosis. But at the moment, it's very much based on the clinician's opinion, and there are a number of tests you can do, but it's more of like an examination, as in they'll ask you some questions and ask you to recall things and ask you to do some perhaps a drawing challenge, and so the diagnosis is currently done in quite a subjective manner, which means diagnosis actually happens quite late into the disease development. What scientists currently think at the moment is that the initial mechanisms to the disease probably start about 20 years before the first symptoms start to be apparent.
Oh man.
So actually, when the symptoms start to show, the disease is pretty well established,
Yeah.
and it could be very challenging to treat this. So if we can spot any patterns in lifestyle and genetics and environment, there's a number of real big advantages to that. First, maybe we can come up with like a diagnostic test that can diagnose a lot earlier. And if we can diagnose it earlier, perhaps we can intervene earlier.
Yeah.
And also, perhaps then we could recommend, say there's a lifestyle event that we think, okay, that really increases someone's chance of getting the disease. Perhaps then we can start to advise and say, look, if you change that lifestyle, then you can delay the onset of this disease. And because Alzheimer's disease is a disease that's associated with aging, it's not necessarily about finding a treatment that can cure it. But if we can find a treatment or an intervention that can delay it by 15 or 20 years, then perhaps we can then push it to a point where it actually doesn't impact person's life that much. Because if currently someone's developing it at 70, if we can push that back to 90, that's a real good extension. And then obviously the risk at 90, other diseases may be playing a part as well. So there's a lot of interacting benefits to really finding out what causes the disease, and if we can actually intervene at an earlier point to delay that disease.
Kind of like when we learned that smoking cigarettes led to lung cancer and stuff like that. Scientists could start advising people to stop smoking because it leads to that. Exactly that.
And there may be some environmental factors, there may be lifestyle factors, and it may be a combination. It's a lot more, I would think it's very likely that it's a combination of everything that we do.
Yeah. A nice little balance between everything.
Yeah.
I think unless, Luke, I'm going to give you, final shot, final 10 seconds of fame. No, I'm sure you'll have lots more seconds of fame in your
if there was
life.
anything more about what you wanted to talk about that you wanted to get out there that we haven't gotten around to asking you.
Now's the time to shoot your shot.
Yeah, sure. So, I mean, often the question is like, what can we do? What can we change in our current lifestyle to prevent dementia and Alzheimer's? And I think it's important at the moment is that there isn't enough scientific evidence out there to make that advice, and there might be plenty of blogs and reports online, but just don't read everything and don't sort of worry too much at the moment about, and there's no need for genetic testing and everyone to go out and start requesting and panicking about genes, because we just don't know enough about it at the moment. And that's exactly the work we're trying to do, and trying to find out what causes it, and hopefully in time there'll be concrete advice of how to slow the progression of diseases such as Alzheimer's. So I'm always asked, like, what can we do now? And at the moment, they're just the basic advice you get from the charities and from experts in the field is try and live a healthy life like other diseases, eat well, exercise well, don't smoke, don't drink too much. It's the usual kind of advice at the moment until we can find some concrete evidence to say different.
Amazing.
There you go.
Thank you so much, Luke, for, sharing your time, coming on our podcast and, doing not your first but your second Pint of Science, kind of, talking about your stuff for the crew. Really appreciate it. And guys, if you really liked what Luke had to say, you want to find out more, you can follow him on Twitter @Luke Whiley. We will share the handle in our description of this podcast and also on our social media. But like I said, he's at Murdoch University. Follow them. The Perrin Institute is also another institute that you can find on Twitter if you're really interested in this sort of research. And like I said, you can Google on YouTube, look up on YouTube the Brain Health in Our Modern World panel discussion that Pint of Science held. But I think that rounds us up, winds us up, wind/round. It turns into rind. I'm thinking
Wounds
cheese. Rinds of cheese. I don't know. I bought this nice like triple brie cheese from the supermarket yesterday. Not the point. but I'm thinking about cheese now. No, that winds up our 2nd out of 3 Pint of Science episodes for this podcast. If you love our podcast, as usual, you can find us on Twitter at CuriosityRat, or on Instagram at CuriosityRat, or on Facebook you can find the page Curiosity Killed the Rat. In future episodes and as normal episodes, we have our listener questions. So if you've got any question about anything science related that you want me to try tackle in a future episode, chuck us an email at curiosityrat@gmail.com. But in general, you know, thank you for coming. Thank you for listening. I hope you guys enjoyed it. I hope you learned something. You know, if you have any more questions.
Thank you again, Luke, for coming on. Appreciate it.
Thanks for having me. It's been great, yeah.
And thank you for Pint of Science. And, you know,
catch you all next week.
Curiosity. Kill the rat. Curiosity.